recover, you'll be fine, and then it'll come back. It's a up and down condition. It can go on for months, it can go on for years even. Again, good chance of cirrhosis developing or cancer. No vaccine is available. Any questions about hepatitis?
Is everyone considered hepatitis because it's related to the liver, or is it, I mean, they're all...
Yes, because they affect the liver. So that's basically, hepatitis is inflammation of the liver pretty much. And we just mentioned 3. I believe that there are 3 more strains that have been identified, ED and F. And I'm not sure how virulent they are, or what conditions they cause, or how prevalent they are, but these are the 3 that your book talks about. But yeah, it's because, not necessarily because they're related, just because they all affect the liver. Okay? And we need to keep that in mind. It doesn't mean necessarily that there's a relation between the viruses.
Right. I just read that Hep C is the highest in Monterey County right now.
Oh, really?
Right.
Yeah.
Because of drug use.
Drug use.
It's also widespread.
I have a question. Okay, so I went to a hospital and they always test for hepatitis, but I don't know which one it is. Like they put like
Probably hepatitis C.
they put like a bubble on your hand. Is that no? They test you for something else. And
If you work in healthcare, like I worked in healthcare for a long time, and they made me get you had to get vaccines, you had to be vaccines.
okay.
And then you had to get the bubble test, which was TB. But if you go back and tell me I don't have a vaccine records, they'll mark a plate on me for that baby. That might be what that is.
Could be, yeah.
Yeah.
So briefly here, let's talk about a couple of microbes that infect wounds. We've already mentioned 2, Staphylococcus aureus and Streptococcus pyogenes. We could also throw in there Staphylococcus epidermidis as an opportunistic infector, if you will. If you remember those 2 microbes, aureus, Staph aureus, and Streptococcus pyogenes cause impetigo or pyoderma, which is an infection of the skin, causes a lesion to occur. You can get pus forming. But the ones that are of major concern are the skin infections that are caused, or I should say the wound infections that are caused by anaerobic bacteria. We're talking about then those microbes that we find in the soils, specifically Clostridium strains. Clostridium tetani, microbe that causes tetanus, produces a exotoxin called tetanospasmin, which actually blocks the actions of the neuron. So it inhibits the neurons. And so the muscle becomes locked. Familiar with the condition of lockjaw? This is an example of tetanus here. When you step on a rusty nail or something, the reason that you have to get a tetanus shot is because of this microbe being in the soils. It has nothing to do with the nail or the rust on the nail, which was always the case when I was growing up, when I was playing in the lumber yards, and I did that a lot. Anytime we stepped on a nail or something, my mom would always ask if it was a rusty nail. Like it really mattered. It didn't really matter. If the nail was in the dirt, you were probably exposed. So, it doesn't have to be rusty. It just has to be something that can penetrate deep into the skin and then introduce the microbe. Once it's introduced into the skin deep like that, the environment is anaerobic, and that allows for the microbe to grow. Clostridium perfringens. This microbe causes gangrene. It grows well in dead or poorly oxygenated tissue. It too produces a toxin. This toxin attacks the host cell membrane. And it was one of the conditions that soldiers of the Civil War had to deal with. A lot of times the bullets didn't kill them. They just introduced bacteria that would eventually kill them, or at least cost them a leg or an arm. Any questions? So that's our semester. That's all I'm going to lecture on. We could start review tonight, or we could save it for next time. It's up to you. A couple of things. This has been a little bit of a different semester for me. Things change, and Hartnell's not very good at evaluating the classes and the instructors on a timely fashion. Last time this course and I was evaluated was 2010, spring of 2010. Since then, I've changed a few things that I've done, and the camera has been introduced this semester. And I'll be honest with you, it had a little bit of an effect on me. Certainly you don't want to become that YouTube viral video that everybody's watching the next day. So I think maybe it tightened me up a little bit, but one of the things I want to know is whether or not you guys actually went and watched the videos and if they were helpful. I mean, some of them say it had 52 views. Well, I don't know if that meant 52 of you watched it or if my mother watched it 52 times. So it would be useful for me to know if that's something that I should keep doing or if it's something that, just makes me nervous and isn't worth doing. A couple of other things. I'd like to know how you feel about the lab, the identification of the unknown. I know you were asked to do so in your presentation, but I'm kind of curious as how you felt about that process and the presentation. That's new. And the YouTube videos. I've introduced those as of recently, and I haven't used those in the past. So I'd like to know how you feel about those. And then one last thing that I'm thinking about doing next semester, and I don't know if this would have been helpful to you or not, but establishing a Facebook page that will allow me to interact with the students in a timely fashion. Because there were a couple of times this semester where if I could have sent out a message to you guys, you would have all gotten it, and it would have been helpful to me to have done so. For instance, there was a time when the lab books were due, and I changed my mind on collecting them, but I couldn't get that to you until the next time we met, which would have been nice to say, hey, don't collect these, or I'm not going to collect those. So if that would be something, I don't know how many of you have Facebook. About 2 years ago, my cousin calls me up and says, hey, you gotta sign up for Facebook and go look at my pictures. So I signed up and I went, and he had one picture. And then he's never on it. So anyway, I like to think that, and I think this is the case, social networking is exploding. And it's going to become the way that we communicate in a lot of ways. I'd like to find a way to incorporate that into the class. I come across papers all the time. I can post papers to it. You guys would get those immediately. The current events, instead of having to write the title and stuff up here, I can send you links to the current events. So things I'm considering on working on over the summer, I'd like to have your input on that. So what I have done to that end is I've put together a little evaluation that I'd like you to fill out for me. Take it home, answer the questions as best you can. I'll give you 10 points for it, and you can do it anonymously. Here's how it works. Don't put your name on it. Don't need to have your name on it. When you come in to take the final, you come in, you hand me your test sheet without your name, or excuse me, your evaluation without your name, and I'll initial your test, and that tells me that you handed it in, and you get 10 points for doing it. Okay? Just a couple of things. Like down here when it asks about the instructor, be specific with your criticisms. Don't say he sucked and just leave it there. Tell me why. Tell me why he sucked, and that'll be great. And hopefully I'll incorporate some of this into next semester, make it a lively semester. Anything else? Be sure you grab one of these before you leave. I'll have 2 piles out here. Well, I think people want to. It's up to you. I have to be here either way. So. Okay? If you don't want to stick around, I don't want to force you to stick around. You're welcome to check out. I'll even turn around. Okay, so we started this section. We were talking about epidemiology. So, if you remember this chapter here, epidemiology was a discussion on the distribution of health state, both negatively and positively. And this is where we introduced the notion of communicable disease versus a non-communicable disease, and the sequence of events that have to take place for transmission. So there's the reservoir, where the microbes are. They have to leave that reservoir, and that exit is generally appropriate. You know, if it's a respiratory microbe, you probably have to sneeze, cough, or get rid of it that way. If it's a digestive microbe, fecal route, or via vomiting, or in the mouth probably. So leaves appropriately, but then also has to infect appropriately, has to enter the body appropriately. So we talked about that. Respiratory microbes enter through the respiratory system. Digestive microbes enter through ingestion. A couple of terms were thrown out. You need to know them. Rate of disease, attack rate, morbidity rate, mortality rate, incidence, prevalence, endemic, epidemic, outbreak, pandemic. All terms that hopefully you're somewhat familiar with, you've encountered before. Know how they apply to bacteriological speak, if you will. We talked about the different types of reservoirs that microbes will find themselves in. There's the human reservoir, probably the most relevant to us because that's the one that we're concerned about. The non-human reservoirs, now we're talking about humans, and it's with these non-human reservoirs that we experience zoonosis or zoonotic, where you have a transfer of disease from animal to human. Environmental reservoirs as well, soils like with Clostridium, and water like with Vibrio. Talked about the types of transmissions, horizontal and vertical transmission. Of course, vertical transmission is just from mother to fetus. Horizontal transmission can be from me to you. And we talked about the different ways that can happen. There's direct contact, where I can walk up to you and touch you and pass it on to you that way. I can get close to you and I can cough on you or sneeze on you, pass it that way. That's droplet transmission. Or I can just be dirty and I can touch a bunch of different things and then transfer it to you that way. That would be indirect contact. And when we're talking about indirect contact via an inanimate object, the inanimate object is a fomite. Other means of transmission, food and water again. One of the concerns with food transmission is this idea of cross-contamination. Air, also a means of transmission. In this case, remember the notion droplet nuclei. The fact that you can have microbes suspended in the air floating around on dried sputum that can then be inhaled. We have vectors also that can serve as a means of transmission, both biological vectors and mechanical vectors. A couple of things to keep in mind about the spread of disease, the factors that can influence it. The dose is very important. Microbes that require a small dose to cause infection probably can spread more easily, or more easier than I guess, microbes that require a large infectious dose. And a lot of times, the dose has much to do with the microbe's ability to avoid the host mechanisms. Incubation period can influence the spread of disease. The longer it incubates, the greater the chance that you can come in contact with more people, spread it to those people. If we know you're sick, we can quarantine you, and we can avoid coming in contact with you. Those diseases that have symptoms right away are much easier to control.